hand over to you, Tamica, and I'll keep an eye on the chat box.
So thanks, Gary. All right, so I'll share my screen here. Okay, so at this point, there should be complete visibility for you guys. In the center of your screen. Gary, can you confirm that so that I know that
I can confirm it. And now if you want to focus and you click on those three dots, you should be able to click on focus on content to make it bigger if you want to.
Okay, all right. All right, well, good day, everyone. Thank you for coming to the webinar and joining us to learn a little bit more about our program and our ELISA kit in particular. My name is Tamica Stubbs, and I am essentially one of 4 training specialists in the Bio-Rad Explorer program. Should you have questions about this presentation or any of our resources, feel free to reach out to me and or my colleagues at the program's email that you actually see at the foot of this particular slide. All right, just for orientation sakes, for those of us who are not familiar with our program, the Explorer program essentially is a collection of mostly biotech-based kits and resources dedicated to making biotech learning accessible to science educators and students alike. The program is a part of Bio-Rad Laboratories, which is a larger commercial entity that produces and sells quality instruments and reagents to a variety of life science-based groups. All right, and so we are a small part of this, a small with a big impact, right, for science educators here in what we classify as the life science education group. All right, so today's, kind of jumping in, today's presentation will serve as an introduction to one of our most popular kits from the program's portfolio, the Elisa Immuno Explorer kit, as Gary mentioned. This will be broken out into 5 major talking points, including ELISA basics, real-world applications and scenarios, which is really the highlight of this kit. It's very, very exciting. And then we'll start breaking out the kit's contents, a little bit more of what it is that you want to know. The student protocol, so we have some experience or at least an orientation towards the student-facing side of things to help with your prep. All right. And then we'll dive into the most extensive part, which is the ELISA kit prep and the resources. Now note that you may actually see a bar pop up on some of the slides here, and it will list out tips and tricks. And those are just kind of, little things that we've learned along the way to help you with your prepping purposes. Right. All right, so as I transition from one slide to the next, I will ask if there are any questions. We'll take a moment to pause here. So we'll start here with the introduction. Are there any questions about the overview and or my position and or how to get in contact with me before we kind of dive into this? Thank you for responding in the chat. I really appreciate that. All right, to start, I guess the very basic question is, what is an ELISA? All right, so an ELISA is short for enzyme-linked immunosorbent assay. The assay in and of itself captures and immobilizes reagents using high affinity antibodies in color development reaction in the final step for validation. So essentially, again, we are using antibodies in a color development reaction to validate that what we target or desire to identify is there. All right, with that in mind, all right, it does detect specific antigens. So it's great for acquiring a specific molecular target and is capable of both qualitative and quantitative information or readouts. And for this reason, or for the many reasons just stated, it is a widely used diagnostic tool. All right, so students will have an opportunity to run a test that's common amongst, clinical diagnostic groups or research groups and get to learn the science behind it. And that in and of itself, while the name of the presentation is not geared towards, the immune system, et cetera, it does relay some information about the specificity of the immune system and how we've been inspired about the capacity and the capacity of our immune system and capitalize on that and box it into a scientific tool. All right, so to run a successful ELISA test, there are 3 key players that are needed. One will be the antigen or anything that triggers antibody production. And so we have this square here to as a generalized representation of an antigen, right? And then we would need an antigen-specific antibody, all right? And I'm using this little green bracket here to show that within this area of the antibody that it was designed specifically to target and identify that particular antigen. And so generally, and so that, an antibody is just a neutralizing agent and it's configured specifically to identify said antigen, right? And so we have those 2 components. And then the third component that we would need, and these are not organized in any form or fashion as of yet. We'll look at some of those configurations here shortly to see how it actually works within an ELISA. Okay, so the third component is an antibody that's conjugated with an enzyme, and that's critical because that enzyme then is presented with its substrate, and then generally the substrates, be it artificial or natural, are generally gives us some sort of calorimetric readout, which is the essence of the ELISA. The response then is colorful and it validates everything that you did prior to introducing that secondary antibody or antibody. All right, or antibody that is linked with that enzyme. All right. All right, so there are 4 major types of ELISA tests. So how we arrange those components that I just pointed out here of those required entities are a matter of what it is that we're trying to identify. While there are a number of ELISA configurations, because there's much more than what you'll see actually presented here, there are 4 common types that are that are employed. All right, so the very first one is a direct ELISA. All right, this is where the wells, which we'll talk about the composition or the chemistry of the wells that are used to conduct these tests, is antigen coded. So there's an antigen coded plate, and we're screening for an antibody. And what that looks like in essence, because it is called a direct ELISA, is that the antigen of interest, we use an antibody that is conjugated with an enzyme that directly makes contact or identifies or targets that particular antigen. And then that enzyme is introduced to its substrate. And then there is a calorimetric readout, which we'll look at different colors that are employed in ELISAs as well. So second, there is the indirect ELISA, which is an antibody capture. All right, we have here an antigen-coded plate, and we're screening, and we're using antigens and antibodies to kind of screen for that particular target. So again, this is kind of an indirect way of going about it, where we have a primary antibody connecting to or binding to the antigen. And then we have secondary antibodies binding to multiple sites of the primary antibody, again, using a conjugated antibody. That enzyme then is introduced to the substrate, and then there is a calorimetric readout there. Third, there's the sandwich ELISA, right? And this is antibody coded, right? And we're using a screening antigen or antigen capture here. So again, here you have the antibody presented first, antigen second. You may have another antibody that identifies the antigen there, and then a secondary antibody or that's conjugated, and then there's an introduction of substrate here. And you get your calorimetric readout. And then finally, one of the most frequently used ELISAs is called a competitive ELISA, where in antibodies, you have 2 competing antibodies and or antigens from a patient, let's say, for instance, that is competing for that particular antigen. That way we know is either present in the system or not. And so the fact that we actually get a calorimetric readout or not will dictate which one of those antibodies had an opportunity to attach to the antigen. All right. So prep contents of this kit will provide and prepare students for the indirect ELISA. So I wanted to make sure that I drew your attention to that. And you can drop this in the chat here, but are there any thoughts on why the indirect ELISA is a more ideal setup in the classroom?
Nothing coming in the chat. I think they were just listening intently and learning, Tamica.
Okay, so better results, easier to read. And yeah, you'll get better results. And for that reason, if you kind of just, hone in here, you'll notice that that secondary antibody, right? That conjugated secondary antibody is binding to multiple places on that primary antibody, as opposed to just the one, right? More stable, less interference, right? And therefore, if you have more of these secondary antibodies binding to various sites on a primary antibody, you have more enzymes available to interact with more of the substrates, and therefore the readout is a lot quicker as well, right? So that's ideal for a classroom, because one thing that we're short on for the most part is time, right? And so we want to make sure that we're able to get results within a timely fashion. And so later we'll take an interactive view of what an indirect ELISA looks like, and that will show us some of the molecular stacking that's actually happening there. All right, so like the assays configurations, there are a variety of enzymes, or sometimes referred to as labels, in corresponding substrates that are used for colorimetric detection. So for example, we actually may use the enzyme horseradish peroxidase with the substrate TMB, or tetramethylbenzidine. Once those two interact, you get I would say a teal-like colorimetric readout. There is also something commonly used, another enzyme commonly used, beta-galactosidase, the substrate O-and-PG, or O-nitrophenol beta-D-galactopyranoside. Once it interacts with its particular substrate, you get a yellow readout. Or you can use alkaline phosphatase with the substrate BCIP, which is 5-bromo-4-chloro-3 idophosphate. And once those two interact, the colorimetric readout is more of an indigo kind of response. For our assay in the ELISA Immuno Explorer kit, we will be using horseradish peroxidase and TMB, so therefore you should expect a teal-like readout. All right, so I alluded to a little bit earlier that we would get to understanding where we're capturing all of this business or information, right? All right, and so we use something called polystyrene, and they're set up in tidal well strips, so it looks a lot like this, and all student stations will have this, right? So our tests are often carried out in polystyrene wells, with the exception of LFAs, which are lateral flow assays, and that's not included in the kit, but that's just kind of giving you some sort of information there, and that's conducted on a nitrocellulose membrane. And so this one is preferred in the lab for this particular setup for several reasons. One, so I'm enhancing here the well to show or to demonstrate what actually happens in each individual tidal well strip, but having a look at why this is so popular. One, it's used to immobilize reagents by adsorption, right? And it's based on its chemistry that we're able to quickly allow for this process to happen, right? So let's say we're leading with an antibody here in this particular assay. And in order for the stacking of the validating or successive reagents to actually occur, to be successful, adsorption has to occur between that plate's interface and the first agent of entry. Now, again, this doesn't have to be an antibody. This is just demonstration here. It could be an antigen like the one we're actually using in this kit. And how this is actually achieved is that the polystyrene in and of itself is made of polystyrene. So we can count on with the specific antibodies to bind to the antigen, but how do we get that antibody or antigen to stick to the well in and of itself? All right, we're going to count on those hydrophobic interactions between the surface and the hydrophobic domains of the proteins. So I don't know how well you can actually see this. I try not to, make this too large here, amplify it too largely, to kind of give you a sense of the chemistry of polystyrene, its hydrophobic nature, and how that then lends itself to hydrophobic interactions between protein domains and the material itself. All right, and so this is going to help to immobilize these materials and allow proper stacking so that we can validate what it is that we sought out to target is targeted. All right. All right. Also, polystyrene is porous, right? And so while that's kind of not a benefit, it does help to teach the science behind specificity. And it does help with validation of the specific stacking, because we actually use soap. Proteins could actually be used like gelatin, but to let's say, to block those particular areas. So let's say these are the excessive or non-bonded pores after introducing that particular antibody. In order to block that, you guys will be preparing a wash buffer with tween in it that then prevents any other bonding or non-specific bonding, so that we don't have negative or false positive results there. All right, and again, we're then able to kind of with certainty say, well, because the other pores were blocked, we know that what was there before we actually blocked those pores captured that antigen or antibody in successive reagents, therefore, and then the readout is valid. All right, so before we move along to the next talking point or discussion point, I'll pause to ask if there are any questions. All right. All right. Thanks so much, Joanne. You're very, very verbal in that, vocal in that chat. And thank you, Tony. All right. So you guys, when I was in a classroom, literally this was my favorite Bio-Rad kit. And the most exciting and I would say appealing part about bringing ELISA into the classroom is the sheer number of simulations that just can be contextualized with the same or similar setup of the provided reagents. So some examples that you can kind of contextualize here, by no means are you limited by what I'm displaying on the screen, but this is to give you an idea. Here are things like GMO testing, right? Testing for proteins that are being expressed in a genetically modified organism, right? That could be used as the antigen, et cetera. Something that's very familiar to all students, at this point, COVID and or even kind of bringing up the flu season, right? Testing for these particular agents of infection. Hormone testing, right? This is really the science behind pregnancy tests. And we've kind of capitalized on that to have conversations about animal conservation. We have a complete curriculum designed around students using the ELISA to test for or simulate testing for hormone, pregnancy hormone and ovulation hormone in panda bears, to show how we go about conservation efforts and how clinical tests such as the ELISA are critical or paramount in that process. All right. Antibody response tests. Students might be very familiar with this now, right? Because COVID kind of brought all that to light, in addition to, what is PCR? All right. Drug screening and testing, right? So they might be interested in knowing how that kind of goes and how that happens, job qualifications, et cetera, using ELISAs. And then all of the ones presented here and more are generally the sciences utilized to collectively create what we call an LFA, or lateral flow assay, where we use the antibodies similar to the ELISA or like the ELISA to generate a calorimetric readout, generally in response with the lines of the single or double lines to confirm or deny the presence of a particular agent. All right, so as long as a desired target has a unique molecular signature, especially proteins, ELISAs are ideal for qualifying that information. When we cover prep, I will briefly walk you through the labeling and reagent specifics for these varying contexts. All right. All right, so I'll pause and ask if there are any questions. I know that was, very short. just wanted to kind of give you an overview of how this could actually be used. Thanks again, Joanne. All right, so let's go into what the contents are in the kit once you actually receive it and how you go about storing it before prepping it for students. All right, so here you see bulleted a list of the kit's contents. Now, I won't read all of them to you, but would like to emphasize two additional favorable features of the kit, including one, the number of students served from a single unit. So if you have a look here, you'll see that, you know, a good deal of kits generally accommodate about 8 student groups, maybe 10. This one actually accommodates 12, right? And so that's a good number of students to accommodate, and that's with 2 to 4 students working per workstation. A second and truly beneficial piece is that this kit is a turnkey kit, meaning that all that you need essentially is in the box, with the exception of some distilled water and some flasks or cylinders to prep the materials. All right. And since students simulate transmission and testing and tracking of agents in this particular kit, most instructors and lab techs are interested to know if the reagents that you see presented here are real, and in fact, they are. So let me take you very quickly through what is in the kit. All right, so we're using real antigens and antibodies, none from humans, however, right? So more specifically, the kit is actually using purified gamma globulin protein from chicken egg yolks for general representation of an antigen. All right, this protein is purified and used to stimulate the production of the primary antibody that I shared earlier in the scaffolding piece or stacking, reagent stacking piece. And so in order to generate the production of that particular antigen is introduced to a rabbit, and that produces the primary antibodies that we actually need. That primary antibody is then purified introduced to another animal to produce a secondary, what we classify as the secondary antibody against that particular primary antibody, so that we know that once we introduce each reagent, that stacking will occur because the antibodies produced in these animals are specific to the reagent prior to the introduction of each one. So will these cause reactions from students? I didn't get a chance to read the rest of that. Gary, if you don't mind.
It was saying, will this cause reactions from students with allergies? For example, if they have an egg allergy.
So egg-based allergies, what you're referring to?
Yeah.
We haven't had that happen. We haven't heard any concerns. We would say at least, at least make sure students, don the proper attire, gloves, et cetera, but there is, no ingestion of the, of the product, and, and it should be okay. Again, we haven't had any issues that have come up with allergies, but that is a legitimate concern.
I'm sure it's a very minute amount which is being used as well, which
All right. So that secondary antibody is then linked or conjugated to an enzyme, in our case, HRP or horseradish peroxidase. And then all of the reagents that you see here that are produced in those animals after being purified are lyophilized, and they come in these amber vials that you see here, right? You then take it from this point, you rehydrate it and then dilute it, which those are steps we'll look at here in just a second, right? But they are shipped lyophilized. All right, so when you receive your kits, you will store the contents according to the instructions on each individual label. So you'll notice that when you're looking at each label, it'll say store at room temperature or, degrees. And so we're just going to literally just take a distilled view of that. So it appears essentially like this. Those items that need to be or are recommended to be stored at 4 degrees Celsius are those listed or more boldly pronounced here on the slide. So that's your antigens, your antibodies, and your substrate. Now, being lyophilized or freeze-dried, your antigen, primary antibodies, and secondary antibodies can be left at room temperature without compromise, but it is recommended that we store it in the fridge, which then leaves the remaining components, right? Which are your plastics and then the solutions required to make your buffers. They are fine at room temperature, your microplates, plastics, etc. All right. All right, so another set of frequent questions regarding storage are those revolving around the content stability in those those two specific prescribed conditions. One being when you store it at room temperature, all right? How long will it last? What might be the shelf life at that point? All right, so the unprepared reagents, your lyophilized reagents at room temperature are good for up to a year. You're prepared reagent like the substrate, so that's nothing that you are preparing. It comes already in liquid form. You don't want to leave that at room temperature but for so long, so we recommend no longer than 2 to 3 weeks. All right, and then those items, if they are stored in the refrigerator or at 4 degrees Celsius, the unprepared reagents, again, those that are lyophilized are good for more than a year, and then your prepared substrate, the liquid, is 1 year at that point. So there's a difference there in terms of stability, particularly where it comes to the substrate. All right, so before we jump into the instructor and lab tech facing side of things, I thought that taking a moment to kind of give you guys a moment to internalize the student facing side might provide you with some insight, maybe help you to be a little bit more cognizant of the lab's needs and consequently feel a little bit more prepared and comfortable with the prep or planning to prep stage. So I'll pause for questions from the previous slides or the previous talking point before proceeding on to this section here. Awesome. All right, so based on, again, we can get a ton of simulations out of this, right? Like, so some of the real world applications that I talked about, those all can be simulated, right? So based on the simulated context, students may engage in one or more of the following protocols that I'll present here. All right, and all of these are highlighted in the KITS manual. Again, this has all been distilled. It's a little bit more detailed in the manual. So great question, Tony. There are student sheets in the kits, including pre-assessment questions, lab tables, and we'll talk about the models. It doesn't come in the manual, but we'll talk about student models in a moment, as well as the post-lab questions. So yes, all of those are included in the manual for copying to provide to the students. Excellent question. All right. So the three main protocols that I'll highlight here, protocol one, and this is protocol one is the ELISA for tracking disease outbreaks, wherein students exchange simulated body fluids, and then you perform an antigen-based test. We also use the antigen as the positive control, and then we track disease transmission. The second protocol is just the antigen detection ELISA. This detects a specific antigen. Again, this is kind of up to you. This is where you have free reign and creativity to kind of decide what we want that antigen to be. Right, and then protocol 3 is the ELISA antibody test. This is where you diagnose exposure to a disease, test the presence of antibodies, and then the antibody here is the positive control. So with us changing up some of those positive controls, obviously the way we go about labeling the tubes that will contain these contents will be different. And all of that is outlined, I would say generously, in our manual. So again, this makes the kit highly flexible and adaptable to apply to current topics, especially disease. The variety of protocols opens the doors, I think, to a variety of simulated storylines and a variety of student roles in these scenarios, including, being lab or clinical text, right? And so what we have found that this, this multitude of storylines has increased engagement with the lab in and of itself, making it one of our popular kits. All right, so the timeline is pretty simple and can range anywhere from 1 to 3 days based on the protocols that I just described to you. And this is based on a 50-minute instructional block or session. And it is also contingent or depending on a protocol selected for implementation. So again, it could be anywhere from 1 to 3 days. Now, protocol 1 here in the tip and trick box, or disease tracking simulation will require more time, obviously, because you need time for the swapping of materials, the uh, the lab in and of itself, and then to analyze the results and tracking things, right? But generically, we could see sessions kind of broken out into pre-lab activities, right, which includes paper modeling or discussion, setting the context, some of those pre-assessment questions that was asked about that comes directly from the manual. Session 2 is where you actually run the wet lab, right? It'll take up to about 50 minutes because there are 5-minute incubation periods, which we'll see here shortly. In session 3, analyze the lab results. Again, this all might be truncated and or just kind of like, a la carte, depending on what scenario or context you're implementing. All right, so I alluded to this a little in the previous slide. Should you want students to have some sort of modeling experience, and if materials for modeling falls into your wheelhouse of responsibility, we have a complete set of student-facing materials on our website that teaches them about the basic antigen antibody interactions. You see here in how the ELISA test in and of itself capitalizes on these adaptive processes. The corresponding paper model figures that you actually see here are on a separate sheet. They can be printed, cut out, and then used for modeling the ELISA scaffold either prior to the wet lab or during the incubation periods of the wet lab. All right, so here I wanted to give you a little bit of a break from my voice, but to also give you an opportunity to have a look at one of our resources. All right, so this is an animated overview of the student lab. This could be used for students and or teachers to provide them with an overview of the steps and give them some insight on the particular steps of the protocol. But for our purposes here today, we might be able to use it just to familiarize ourselves with these steps within the lab, which in here, protocol number 1 is going to be modeled, and to inform us of our collective prep efforts. So I'm going to go ahead and play this. It's about 4.5 minutes long. So I'll start this so you get an opportunity to see what the lab looks like in action. And I'll pause there. My apologies. I just want to make certain that everyone's able to clearly hear the
Yes,
audio.
volume is working, yeah.
Oh, fantastic. Great. All right, so that is essentially the overview of the lab. Again, hopefully that was helpful for you to kind of give you an opportunity to think about, what the setup may actually need to look like. Now, this one won't take as long. I did want to give you an opportunity to actually see what the results look like and, at the final step in real time, right? And so here is the qualitative ELISA, and we'll look at it from the substrate forward, and it gives you an opportunity to um, see just how fast the color development actually happens, which would of course attenuate depending on when you prep your materials and how long you allow it to sit prior to setting up student stations. All right, so with that in mind, all right. And so the incubation would happen for 5 minutes. And again, you can see how rapid that color development occurs. And you also may have noticed that the protocol is actually performed in triplicates, right? There were 3 positive controls, negative, etc. And this is, for validity, right? This is for to validate that your results are correct. When you're assaying the samples in triplicate, it is yet another control, right? It gives reference points for students to take note of their particular samples, which are unknowns at that moment. And obviously, if you don't get the same result in all triplicate wells, you may have a problem with experimental technique and or pipetting error. And that actually helps us to kind of zero in or troubleshoot what may have occurred there. All right, so notice in the student lab protocol, the incubation and washes, and I won't get into too much detail here. Just a general note that the 5-minute incubations, that is an optimized time point at room temperature for proper absorption initially, and then binding of the antibodies as well as color development, even though that can happen a little bit faster. In terms of color development, if you want the most accurate results, you want to get the readout within the first 5 or 10 minutes. Otherwise, you actually may be looking at some non-specific bonding results and what have you. And we just want students to be able to be clear on their results. And that the tween wash buffer, right, that's added to prevent absorption or non-specific absorption of materials that succeed the entry material, right? Either the antigen or the antibody. All right, and so asked earlier, right, are there sheets for students to record their information? Absolutely, right? And so here, because there are 3 different lab protocols, we do provide different worksheets to address specifics within that particular protocol, because again, you might be identifying an antibody or targeting an antigen in particular. And so the questions are designed to help students think around that particular ELISA protocol. All right, so before we get into the meats and potatoes of the lab tech side of things, I'll stop and ask, are there any questions regarding the student protocol? All right, fantastic. All right, so how then do we collectively pull these experiences together? Before you, right here is a recommended prep outline table with optimized timing for reagent prep. The caveat I will say is that there are some points of flexibility. This is just not hard line, as you see here, as the components of this lab can be prepared and remain stable for up to 1 or 2 weeks prior to you setting up the student stations. But this is what's recommended for a more optimized setup. All right, so breaking out each one individually, this is what the part that you're keenly responsible for, and that's prepping the lab. So this has to start with the buffer prep itself because they are required. All right, so I'll back up. I believe the question, can you read the question to me? Because it disappeared before I had an opportunity to read the entire thing.
The question comes from Tony, and it says, the substrate color starts immediately. How do we explain this to the students?
So again, we're performing an indirect ELISA, and if you remember just from the graphic, the primary antibody connects to the antigen, but then those secondary antibodies have multiple attachment sites to those primary antibodies, and those are the ones that contain the enzyme. So you have more secondary antibodies connected than you would, let's say, a direct ELISA, right? That means you have more enzyme accessibility. And so once the substrates actually come in, it happens rapidly, or it can happen rapidly. Hopefully that edifies. Let me know if it doesn't. All right, so again, we're going to start with preparation of the buffers, right? Because we will, this will be required to rehydrate our lyophilized reagents. And so this should be at least completed at least a day or two before the day of the lab, again, because it's required to prepare the other reagents within the kits. All right, I'll go back to, I'm sorry, to emphasize that the time required for this, we're reading the color, this means the colors. Gary, can I get you to read out the question again?
Sorry, it says, yes, but we are reading the color. This means that the color is going to be stronger than the last wells.
And so you'll notice it does depend on how fast the students actually introduce the substrate, and it might appear a little bit stronger, but again, in a qualitative sense, as long as there is color development, we know that that's a positive, right? Whatever it is that we were targeting has been targeted, identified, successfully identified, and then the readout. Now, if you're doing something more quantitative, again, you still want to give color development about 5 minutes, 5 to 10 minutes. You don't want to do any quantitative assessment after that, but by then you should have full and complete, similar colors, right? Because once the substrate is consumed, right, it can't develop, a different, more color simply because there's no more substrate to be consumed, right? At some point you run out of the amount of substrate. And so at some point the colors will be even. You're very welcome. All right. So what does this basically look like? You're going to be preparing 2 buffers. The materials needed from the kit are featured on the left, and then the additional materials that are required for this part of prep is indicated on the right, which are graduated cylinders. You'll need one that measures up to 1 liter, as you'll be preparing about 900 milliliters of one solution, and you'll also need something 100 milliliters that can that has a capacity of at least 100 milliliters, because the other solution is about total volume of 100 milliliters and some distilled water. All right, the setup here is fairly straightforward. So you'll take your 10x concentrate of the PBS or phosphate based saline solution, and you will just transfer 10 milliliters of that and combine it with 90 milliliters of distilled water, and then you have your 1x PBS solution. All right, so as noted, this will be utilized to rehydrate your lyophilized reagents and bring them to a 50x stock concentration, as well as being used as materials for our negative samples. All right, so for your wash buffer, which is our second step, we're going to prepare our wash buffer. I know these measurements seem very, very specific, but you'll use the remaining amount of the PBS, which is 90 milliliters, and there's a total of 5 milliliters of the 10% tween, but you only use 4.5 milliliters of that. And again, that's going to be used to kind of block some of those pores in the wells and to wash out remaining agents, and 805 milliliters of distilled water. All right, and this will bring it roughly to about 900 milliliters of total solution. All right, now we will be using the wash buffer to further or to dilute the 50X antibodies and the 50X antibodies alone. And we'll have a talking point about that here in just a second. But it's important to note that you do not want to dilute your antigen with the wash buffer. All right, so the next step, we're going to rehydrate our lyophilized reagents, which are your secondary, your antigen, your primary and secondary antibodies. All right, so this should take about 5 to 10 minutes as well. I'm sorry, I need to make sure that I emphasize that. Kit materials on the left, additional materials on the right. All right, so our prepared materials, the 1X PBS, we're just going to transfer a half a mil or 500 microliters of the 1X PBS solution into each of the vials containing the freeze-dried agents or reagents. Now we can use plastic pipettes from the kit. So again, I discussed that this is turnkey, and so, but micropipettes are just fine as well. It's a matter of preference, but they can be used just as long as that there is no, or there are measures taken to make sure that there's no cross-contamination between the agents as we're transferring over the PBS. All right, the next step is just to dilute your 50X reagents, which was prepared in the previous step here. So that brings it to a total concentration of 50X, those materials. All right, this should take about 10 minutes. All right, so we'll take our prepared materials, again, our rehydrated reagents, our wash buffer, and our PBS, and additional materials. There are transferring tools as well as 3 small beakers to house the diluted solutions. All right, so the initial step in this part of the procedure would be to label 3 50 milliliter beakers or plastic cups that has a capacity of 50 mils or more, and you would label them according to their new concentration, and that's 1X antigen, 1X primary antibody, and 1X secondary antibody. All right, so for the antigen, we'll kind of separate them out because we're using different solutions to dilute them. So to dilute the antigen to 1X status, you will take all of the contents, which is 500 microliters or a half a milliliter to the container, label 1X antigen, and 24.5 milliliters of 1X PBS. And this will bring it total to 1X solution or concentration. All right, for your antibodies, I emphasize here in green and in larger letters here, just to, to iterate or reemphasize the fact that we want to use that wash buffer only for the antibodies. So to dilute them and bring them to a 1X concentration, very similar, 24.5 milliliters, however, a wash buffer here to those respective containers. And again, word of caution, only use wash buffers on the antibodies or the experiment will not work. Then we'll then dilute the contents of the respective vials, 1X primary, the primary antibody to the 1X primary antibody, label container, and similarly for the secondary antibody. All right, so I just wanted to take a moment to pause and get you all to think about why the tip and trick here is critical for a successful experiment. All right, so as a reminder, all right, students will be running an indirect ELISA, wherein the antigen is first absorbed to the polystyrene plate, and then the succession of materials are presented as this as represented in this graphic. So, are there any thoughts to why we, set out this precaution here to make sure that you understand to only use the wash buffer with the antibodies. Why would we call that out specifically? I know it may take people a moment if you are thinking about it, and then to drop it into the chat to type it in. So I'll give you guys a second. Don't know. Very honest. Very honest. All right. To avoid
contaminants.
Contaminants. Got it. All right. So again, remember, there are two purposes of the wash buffer, right? The wash buffer, one, actually washes away the materials or excess materials that we've introduced, those that are unbound, right? So that's one of its functions. And the second function, primarily in that first wash, is that there are open pores here in the polystyrene well, right? And we're using the tween within that wash buffer to block all of that. Now, should we use, let's say, the wash buffer for the antigen in and of itself, right? Then that means that that might prevent the antigen in and of itself from locking on or absorbing to the polystyrene well in and of itself, and therefore this stack or scaffold that you see would never happen. Okay. Right, and so obviously that would ruin the results of your potentially ruin the results of the lab. So we only use that for the antibodies because by then the wash buffer has already been introduced to the wells. All right, so a potential stopping point, you can literally take your reconstituted materials and diluted materials and store them in the fridge or at 4 degrees Celsius to up to 2 weeks before the lab or before proceeding to the next set of steps. All right, and that's dispensing the reagents and solutions and preparing the student lab setups. So what we require for this is the part that takes a little bit more of time, right? So the total prep time is like 1 to 3 hours. Literally pulling together the reagents are several minutes at a time, up to 10 minutes, but it's the labeling that's a little bit more intensive in terms of prep. So we'll need all of our prep materials and additional materials. We'll need cups and something to label our microtubes as well as some cups with. All right, so you'll start here, tubes. And this will be done according to the instructions in the manual, right? And this is contingent upon which contact you're actually using. However, what is consistent are the blue being representing the positive, the purple negative, and your primary, secondary antibodies substrates in these colors. Now, what you might label that yellow tube will be different depending, again, on the context. So this is why it's, sample question mark here. All right, and this will be for each student station. So again, this is why this is a little bit more intensive in terms of on the prep side of things. And then we might ideally want to label the contents of the cups so that the buffer isn't accidentally used prematurely, for the reasons just discussed at the expense of the lab's outcome. All right, again, the lab prep, dispensing of materials, very easy, very direct and outlined in the manual, right? And so for our positive control, we're going to use in this particular protocol, a 0.5 ml for that. And we'll also use the antigen for a positive student sample, again, context specific at that point. And then we'll use the 1X PBS for our negative control as well as negative samples, student sample, depending on what it is that you're doing here. All right, and please note again, kind of highlighting that turnkey piece, that the gradation marks on the plastic pipettes are adequate enough to measure out the liquid volumes required for dispensing these reagents. All right, so it'll take care of those particular colors, and then the primary antibody, 1.5 milliliter, goes into the green tube, secondary antibody into the orange tube, or what's supposed to appear to be an orange tube. And then again, you notice you didn't have to prepare the substrate because that comes prepared for you in liquid form. You would just simply transfer that into the amber tube. And just note that the substrate in and of itself is light sensitive, which is why we put it into the amber tube to protect it. And so it always has to go into the amber tube. So it is light sensitive. All right. You'll want to transfer at least 70 to 80 milliliters of the wash buffer. Students will be using this frequently, as you saw from the animation. And then when ready, okay, again, I said this timeline is kind of flexible. So, but the day of the lab, you want to go and set up stations. But again, the stations, materials and the tubes or dispensing of those reagents can happen earlier on and just set in a fridge for up to a week, and then you kind of take it out. So it kind of depends on your personal and professional timelines. So the total setup for students would look like this, potentially including a rack to hold the microtubes. All right. And again, I'll just emphasize that though this is labeled S1 and S2, that might be different depending on the context. So a tip and trick here as well. What we've noticed as well is that keeping the microtubes in the order of use is helpful for students. So what that would look like according to the labeling of the title well strip is that you'll have your positive first, your negative next. So this is the succession of things, and then student samples, right? And then all of these wells will then receive primary antibody, secondary antibody, as well as the substrate. All right, so the idea of protocol. So what you will label will be contingent upon which context you use, which protocol you use, or which complementary protocol that you actually use. Now, protocol 1 in particular is the where students swap materials. Now, according to the manual, you guys will notice that the second page or the second page presented here of prep will be similar to our protocols. What's different generally is I would say the second page of prep. The first graphic was the cover of the manual. So the second page of prep, and I'll call these things out here. So if you're running protocol 1, you'll notice that here there's a difference in the amount of or the volume of your of your positive control, right? You're using 7.5 mils of 1X antigen. And here at the bottom, it calls out because students will actually be randomly swapping or, simulating the swapping of bodily fluids in vitro, that we would want some of those student samples to be more concentrated than others, because the idea in this particular protocol is to identify patient 0, right? So dispensing different concentrations of samples is key here. So this is what's called out right here. All right. This is then followed by the antigen detection protocol, which is protocol number 2. And this is where they will track down a particular antigen and find out who patient 0 is. Now, this is designed to generate additional discussion about health issues and epidemiology, potentially STDs, depending on how sensitive your clientele is, or, the spread of COVID. So often we actually run a zombie, virus kind of scenario. It's often a fun way to get students engaged with the ELISA protocol, I will admit, right? While potentially mobilizing a fictional zombie virus, it is inconsequential, and students don't feel like, this might be real at this point while they're swapping solutions, as they would if you kind of contextualize it a little bit differently. And so again, we're going to note that the swapping of fluids is recommended for an independent session, because this may take some time for students to exchange fluids and record information. But on a student sheet, there's actually a place for students to record information about who they exchange fluids with. These fluids can then be stored in the refrigerator 4 degrees Celsius until the next day, and then you can conduct the antigen-based protocol, right? Another one, right, or another scenario, should you want students to kind of connect to a more non-fictional context like the spread of the seasonal flu. You might simulate a sneeze or fomite contact with the protocol seen here to demonstrate the virus's passage and test via ELISA protocol number 2 in the following session. All right, so protocol number 2, again, this is the antigen detection. And because I know we are running out of time, I'll just go ahead and say we do have a couple of interactives that shows you what's occurring up close and personal in the tidal wells. So it literally has, an animated version of what it absorbs first, the washing of it, what then, connects or binds to the antigens. And what's nice about this particular animation is it actually shows a collection of proteins that may be coming from the patient sample, right? Because it's not just the antigen, even though we're scoping that out. It does speak to the specificity, however, of an ELISA kit, which is great, right? But these links will be provided to you. Gary will send this information to you guys so you guys can give it a try for yourself. And so that's the antigen detection protocol, right? And again, you can be very creative with your choice of antigen here, but again, you'll follow the protocol that's simulated here or outlined in the manual. All right, calling the call out here in particular for this particular protocol, literally what is noteworthy is just basically the recommendation for the distribution of positive and negative samples. And we say try to kind of keep that equal, keep the ratio balanced. All right, so for this particular protocol, the antigen detection protocol, one option could be confirming a pregnancy in humans. And this one gives students a taste of the science that actually goes behind into the development of the lateral flow assays. So those are the sticks, right? That we, kind of get one band or 2 bands, right? Or the happy face or not, right? And so what is the science behind that is informing or creating those results that we just telemetrically read out? The manual, again, will provide you, as you see here to the right of the slide, with the descriptions, simulated content. So you'll see the tube description, right? What should be contained in there, respective tube colors, what the content of that should be, and then how you might go about labeling it, right? Or at least making students aware of what the simulated information or content or representation actually is there. So that's a cool one as well. All right. You may just kind of leave it open, right? So another option, as I'm presented here, is GMO testing, right? Because there are a lot of LFAs actually used for GMO testing. So for example, testing plant parts that express pesticidal proteins. Smallpox is another great real world example here. And it's particularly useful because if you detect it within 2 to 3 days, it can be treated, or at least during its course. So that's pretty cool for students to kind of be aware of, because then there's a level of urgency, right, in handling their ELISA. So I left this one blank more so to kind of give you the idea that, hey, if there's some antigen you'd like to test and say, oh, this would be a cool one, right, then you would have to kind of go out and find the information and label it respective to that particular ELISA. But again, it gives you so much flexibility and freedom and creativity, and it's really, really cool in that aspect. All right, so again, I won't play this one, but we do also have an antibody specific animated modeling of what happens in the antibody detection protocol. And this is hyperlinked, and you guys will receive this presentation, so you can just click on it and you can kind of manipulate it. It doesn't take very long, but it does actually show how that goes about what's happening in their tidal well strips as they're going through the procedure. All right, a specific call out and protocol. 3 here is that the students' yellow tubes will have primary antibodies and not be antigen, right? Because we're testing for the presence of a particular antibody. And so what might be good ideas to kind of, run this protocol, though it's not as a hot topic anymore, and we talked about curing it in the past couple of past weeks, some revelations there. HIV, right? So sometimes, just the presence of the antibodies or early detection of that gives you an insight to the presence of HIV, because we know that this attacks the immune system in and of itself. And so it can be brought to detectable levels, kind of get students to talk about that. But you can also, use this to detect COVID antibodies as well, or anything that you can wrap creativity around. So that was a mouthful and a lot of information. I do thank you very much for being active listeners, and we'll take any questions if there are any that are remaining or lingering.
Now that was really good. Thanks a lot for doing that. I think Tamica's done a brilliant job there, and also a special thank you to Joe and Tony for contributing in the chat box. It's these presentations aren't easy, and when you can't see your audience, it's always a lot harder. So thank you very much for your contribution. As Tamica alluded to, we are going to share the presentation. We're going to share the presentation, which, sorry, my phone keeps buzzing. We're going to share the presentation which you saw today, and also Bio-Rad do have lots of resources on their website, teaching notes, technician preparation notes, student worksheets, and some nice animations just to outline exactly stage by stage what happens. So I hope everyone did enjoy it. While we're waiting to see if anyone else has any questions, I'll go through a couple of extra education bits, which I forgot to mention at the start. With it being a Breast Cancer Awareness Month, we are supporting a company called Wear It Pink, a charity, and you can buy pink Bunsen burners for this month only. It's a special limited edition pink Bunsen burner, and 100% of the profits go to charity. I'll just put a link in the chat box for anyone who might be interested in that. There we go, a beautiful pink Bunsen burner. This will be our last webinar for this year. We are still going to run a quiz, I believe, end of November, beginning of December, Christmas time, where there will be prizes to be won. So please look out for that. I think you had a teaser question a month or so ago from your local rep. And have we got any more questions coming in?
There was one from Anne. She asked, does the kit contain enough reagents to do all 3 protocols?
So typically we recommend 1, but clever of individuals, right? Because we can accommodate up to 48 students. So that's contingent upon the student group size, right? We could run more than 1 protocol if we're accommodating fewer students, sure.
If once this webinar finishes, you do suddenly think of lots and lots of questions, then please do email us. We'll always get you an answer. Say Bio-Rad are very good for supporting us if you have any questions we can't answer ourselves. Lots of thank yous. Thank you, Anne. It looks like everyone's happy, which is a good thing to make.
Thank you, guys.
So we will also load this recording, which I'll stop now. Now I'm talking. This recording will be loaded